So this is an introduction to lab exercise 20, which is titled Ecological Diversity and Interactions in Plant Communities. And you can see in the background, this picture is taken at night with some night cameras of an endangered ocelot found here in South Texas and thorn scrub habitat you see in the background. And so this structure, the plant community structure here, provides habitat for this ocelot. So there's a specific habitat structure many animals require. So this one is about describing plant communities, analyzing them, and even looking at some of the interactions between plants that occur within these plant communities. So your learning objectives here are to observe physical factors, plant dominance, and interactions among organisms in terrestrial community, and characterize the community according to those features. And then to quantify, which means we're going to put numbers to the distribution and abundance of plants in a plant community. And then to detect the effects of competition and allelopathy. And this third one lends itself to experimentation. And then explain 4 different ways to quantify the importance of different plant species in a plant community using transect data. And so the first activities are involving qualitative, where you would go out into a habitat that you can pick of your choice. And you would do this on your own. And you're not required to do it here, but just to know what should or could be done. And I encourage you to do this on your own. You visit a local plant community, and you're going to go characterize it just based on qualitative nature. That means you're just kind of describing it with words. You're not really putting any objective numbers or measurements to it. And when you do that, you would be observing physical factors, and then you would just go into that habitat and answer these questions about what you see out there. And then you would answer questions about that. So this is your interpretations of what you saw. And then you would go out and look at the plants that are in that area. So first was the physical factors, like is the soil, is it dry habitat, is it near a lake, is it a forest, is it an open habitat? More open type stuff, the physical factors. And then here for your plant, you're going to go look at your plants and try to look at just qualitatively which ones look more abundant within that habitat and so on. So this one's about describing the plants there. And then another little section here where you would interpret what you saw and observed about the plants. It's all qualitative, no numbers yet. And then finally, interactions among organisms. And there you would go and look for signs of interactions, plants with plants, people with plants, other animals with plants, animal-animal interactions. And you write down your observations here. I got a picture of a sunflower, girasol in Spanish. So the species is Helianthus annuus. And I was taking a picture of it because it was just there in the sunlight. And then after I took the picture, I realized that there was a little predator in there waiting for a pollinator to come in there. So a nice little spider. So that's an interaction. That's several interactions that could be described there. And so what you would do is you would answer questions about that. Now the next thing to do is a quantitative community assessment, where you go out and you use a line, a measuring tape, 10 to 30 meter measuring tape, like you see these students here. And you go out and you actually make some measurements on 3 different quantities. One is how frequent you see each plant species. There's going to be several plant species there. Then how many individuals of each species you see. That's a different thing. And then they are different. And then one is how much for each species covers that or touches that measuring tape. How many meters does it cover? And all this is explained in some tutorial videos that take you step by step. And you'll be doing a simulation using an electronic map. And it could also be done if you had a copy of it on top of a table. And basically you're simulating based on a diagram of the distribution of plants within a certain area of habitat. And you go out there and you make these types of measurements. So there's a simulation that's described in other videos, so you'll be doing that as well. And of course, your lab always has questions to answer. This is the procedure for doing the sampling in the plant community. which is described. You're going to be doing this desert habitat, and the map will look like this. And in fact, you can download the map by clicking on the image there of the map, and it'll download as a PowerPoint. And when you open up, you can run the simulation just from your computer there. You don't need to print it up. And I give directions on how to do that through other videos. And then this is data you would collect during the simulation, and it's a simulation of what you would really be doing in the field. So you could actually go out and do the same techniques in the field. In fact, when I teach students face-to-face and we actually get a chance to get out in the field, we run the simulation first so students have an idea of a knowledge of what they're doing when we do get out into the field. And then this table involves calculations that you'll have to do once you've recorded that data. I have a tutorial that explains how to do these calculations. They're also explained in the lab manual if you go through it as well. And then the last thing is to go through the plant interactions, and this is where experimentation comes in. And so you go out and you start to see plants, and you can see signs of sometimes of competition. Sometimes one plant grows high and shades another one, and so it's outcompeted for sunlight. And that's the thing about competition, is it's going to occur. It occurs within a species, but it occurs between and among species. And if it occurs within the species, which have the same kinds of needs, if you're the same species, that's called intraspecific competition. So that's competition within a population or a species, and this happens with animals, and it can happen with plants. And then we look at interspecific competition, and that would be competition for limited resources between species. And so you can see signs of that that suggest out in the field, and then you could design either a field or a laboratory experiment where you would actually control all the factors except some factor that you're trying to test to see. And so one of the procedures, you're not going to do 20.3, or it's not going to be done, but it's within a species. So this would be intraspecific, and here you would look at or could do an experiment. We're not going to do it, but you do an experiment where you use the same size pots with the same amount of soil, and then in each one you're going to have a different treatment. Same soil, same amount of water, same amount of sunlight, but you're going to be putting in one pot only 2 sunflower seeds to germinate, in another one 4, 8, 16, and then 32 years, so you're doubling. So this one might have 2, and then this one would have 4, 8, 16, and then another one with 32. And in a classroom situation, we would have groups, and each group would set up one of each, and those would represent one replicate of each, and then other groups would have the same setup. So we might have say at least 5 replicates of this, 5 of this, 5 of this, 5 of this, and 5 of this. And now we can compare, and the replicates are to repeat the experiment so that we can get a handle on the variation that occurs naturally within these things. So we want to be able to observe that variation and include that as part of our assessment if there's truly an effect or not. These things have already been discussed earlier when we did a review of science methods. And you all have done already a seed germination project that you tested your own factor, and I believe everybody's already turned in their posters presenting their results. And so there would be questions to answer about this. And then there's one other concept, and so little section that describes what allelopathy is. And allelo means other than, and then pathy is you have an effect on it. And so it turns out that plants compete with each other the way bacteria do for resources. And so just like bacteria can develop compounds that impede or inhibit the growth of other microorganisms, plants evolve to do that as well. And they'll evolve these compounds in their leaves and their shoots above ground and in their roots. And so they would release compounds into the environment that slow the growth down. And here you can see, I just picked the first image I can see on a habitat that's dominated by Australian pine, and they appear to release allelopathic compounds. And one of the signs there is that you don't see much ground cover on here, even though there's open spaces where plants should be growing. Sunlight's hitting the ground and nothing is growing in that area. And it turns out that the leaves, when they fall, they have compounds that slow the growth of other kinds of plants. So in this particular one, The lab manual suggests testing a bunch of different plants for this allelopathic compound. So of course you would need a control. So you would need some seed, some plant you're trying to grow, and something easily grown, and then you treat it with compounds that you extract from plant tissues of another plant. And so you would be running a control with just those seeds you're trying to grow in water, some other species, and then one with the plant extract compounds. And so these are at least your two groups here. And then, of course, you need replicates. So during the pandemic that we had, we stopped coming to class after spring break. During the spring semester, I recruited my 8-year-old to help me out. So we actually did this experiment using not pinto beans. We've done pinto beans in the lab before. And we pick a tree that is known to have allelopathic compounds. So we just wanted to test that out. This plant is called Ailanthus altissima, and its origin is from Asia, but it's been brought over here on purpose, and it's very invasive. And it grows rapidly, and it can outcompete native plants for growth, so it can cause damage to the ecosystem. And so we did that, and what we did was my neighbor has some around the house, and it's a constant battle trying to keep it out of the yard. You can see it here growing in the flower bed in front of their garden. So we went and collected some of the tender shoots above ground, leaves and young stems. And you can see there we masked a certain amount there. So we had a little digital scale there at the house, and we got some of the plant material from there. And then we put it in a blender with water, a certain amount of leaf. We put a certain mass to a certain volume of water, and then blended it up. And then you can see the blender makes a lot of noise, so I thought that was a funny picture there. But we did that, and you can see the water has turned green. So we've done that, and that's basically to break apart the plant tissues, try to get the compounds out of the plant tissue. And I thought that it would be good that if we're going to test this solution against water, how would we know it's not just breaking apart the plant itself, that if we do get an effect, how do we know that it's not just the plant? So I looked in the fridge and found some cilantro. And so you can see on this bottom picture here, here's the cilantro there, and then in the cup, and we're masking the same amount. We cleaned out the blender and then made a solution with extracts from cilantro. So we're going to test cilantro, and we tested Ailanthus, and then we tested all that against a control of water. So the cilantro is kind of like a control for hacking up the plants, and then there's the water control. And we didn't know what we were going to get, so we did that. We hacked it up, and we filtered out the water. And so there we have. You can see a picture here, kind of cut off there. There's the water. This is this is cilantro. You can see on the lid, and then the Ailanthus here. And this is how we filtered out the two. So you can see now we actually had some petri dishes. You can use, like you did in your experiment, a plastic bag with paper towel. And we put the same volume. We used these oral syringes to make sure we put the same volume of water, or the water with the plant extract, in each replicate. And so we had 3 groups. We had our control, we had our cilantro treatment, and we had our Ailanthus, known to be allelopathic group. And so when we did that, we ended up with 7 replicates each, and we put 15 radish seeds. So radish was the organism we were testing for the allelopathic effects of Ailanthus on radish. And we didn't stack all of the cilantro together and all of the radish. We mixed them all up to help reduce any issues that can occur with placement of these. So there was a random placement, but we labeled them. Every one of these has a label. You can see a little label right in there. It tells you if it's water, if it's cilantro or Ailanthus, and you can tell which one at least has the plant. We don't know which one it is because the paper towel that was in there looks green tinted. This one's probably a water control right there. So we let them grow. And for much like your germination experiments you've already done and growth experiments and analyzed your data already, we let them germinate for 24 hours, and then we went to look and count how many germinated out of the 15 in each one and recorded the data. Then we did it again 48 hours, And if we still saw a lot of seeds not germinating, we went again and did 72 hours. And then at 72 hours, we went ahead and took some calipers. You can use a ruler like you all did. And we measured the lengths of the roots, or the radicle, which is the young root, and the shoots, and collected that. And we only did that for one seedling randomly picked from each replicate, right? And so that would represent, and so when we did that, we had to have a system for randomly picking the seedlings. And so we came up with this grid situation. And then before we went in there, we ahead of time said, I'm going to place the petri dish on that grid, and we're going to pick from, say, D12 or something like that. So that would have been whatever seedling was closest to D12. And so if that was, let's see, this is D12 right here. And so we would pick whichever one, any part of the seed that was closest. So we might have gone with that one. And that keeps us from purposely picking ones we like. We want a random pick from each one. So we always want random samples. And you can see the difference here. This one, I marked A1 through A7. This looks like it's A7 replicate. And when you compare this to, I think this one is the B one, so the B group was the cilantro, you can see that these seedlings grew much more than this, so we can already see visually there's inhibition, but we always want numbers to support this. And so these represent random removal of a seed from each one, and this is the Ailanthus one. This one, these other two, one I can't recall which one I didn't label, and that's bad of me. But one of them is the cilantro, and the other one is the water control. You can see the cilantro and the water control just visually didn't look the same, but these represent one individual from one of the 7 replicates. So we picked one random one from each replicate. So we had 7 of these kind measured, 7 of these kind measured, 7 of these kind taken from each replicate. And what we did, this was our data. So you already have the data. You know how to design experiments. So you're not writing an experiment. We ran it for you. We were your lab technicians. And here's the data. And I provided the data for you so you can go and practice analyzing the data. You already done this same kind of analysis. Although you only had 2 groups, we had 3. So that's the only difference here. You can see the water, the cilantro extract, and the Ailanthus, and N is the number of replicates. There were 7 each, and this is the germination rates. This first group right here, from the left here, that's the 24-hour and then 48-hour, and you can see that the mean, almost all, remember each one had 15 seeds. The average was 15.6, 14.6, 14.7. and 14.6. Here's the standard deviations. Basically, after 24 hours, almost all 15 seeds in every replicate, with the exception of a few. And the ranges are test ranges following that method described by your lab manual. That's not really a formal test, but where you took the mean plus or minus 1/2 the standard deviation. Those are the ranges here, and they all overlap, so there's no significant difference. And then by 48 hours, even the Ailanthus, the one we're expecting to have an effect, all 15 germinated. In all 17 replicates, there was absolutely no variation, no standard deviation. We couldn't even calculate a range for it. So it seems that the Ailanthus, when we compare to the cilantro and the water, they didn't have 100% of the seeds germinate, but virtually almost all 15 did. And so we would take this to interpret that just biologically, there does not seem to be an effect of Ailanthus on the germination rate of radish. Okay, that's the only conclusion that we can make. We didn't prove it, but we have data that suggests that. But now when we come to the radicle and shoot lengths, and I showed you pictures of those, and you look, the radicle lengths are in orange. And the error bars represent the test range. This is plus 1/2 standard deviation. This is minus 1/2 standard deviation. The middle 2 columns are for the cilantro. Here's the Ailanthus, the one we suspect of allelopathy, and here's your control. If you look at the orange bars, those are the radical lengths, okay? And you can see that the means are never going to be the same. The water looks like the radicals grew longer with the water than with the cilantro. But when we look at this error bars here, these error bars overlap, so we don't have enough evidence to say that the cilantro had a negative effect on the radish growth. We're basically going to say that these two are not significantly different, no effect. But look what happens when we look at the ailanthus. Not only is the mean way down here, but look at the top of the error bar range for our test ranges. That value, that highest value is around 5. The mean is lower than that, okay? If we look at the lowest value on these error bars, they don't overlap with these. They don't have the similar numbers. So that means that the ailanthus radicle length, the radish treated with ailanthus, had significantly smaller radicals. So that would suggest that ailanthus has an effect on growth of radicals. And when we do the same thing with the shoot length, the green parts that were measured, no significant difference between cilantro and water. In fact, the cilantro has slightly higher average, but no significant difference. But when we compare the ailanthus shoot length to both the cilantro and the water, the ailanthus seems to show some sort of allelopathic effect. The shoot lengths were significantly smaller on there. So these are the actual results from that lab, and it was pretty cool to see that verify the allelopathic effect that ailanthus is known to have on other plants. The rest of this PowerPoint are appendices that you can use to practice writing your hypotheses, how to calculate standard deviation, which is an important part of for each group mean. And then here is the significance test that you used in your own experiment and described by the lab manual back in lab exercise 1 for your 1406 class, which says that we're going to judge the means to be significantly different if we take the mean and add and subtract 1/2 standard deviation. We get a high and low range. If those ranges calculated by this do not overlap, we take the means to be significantly different, which is what we just did. Those error bars on this graph represent the mean plus or minus 1/2 standard deviation. So this is your intro to the lab. Basically, we collected the data and these are the results for, so you're going to want to know and review experimental design, how to interpret data, how to assess with the method we're using for significance test if means are different or not between control and experimental groups. But the only other thing is now for this lab is to make sure you go back and go through that map simulation, because you're expected to know how to collect the data for examination purposes. You're expected to know how to analyze the data and interpret it, and that's what the other videos are about. So I'll leave that the rest of your time you're going to be spending with this lab to go back to go look over the videos that explain how to run the simulation. And let me know if you have questions.